I'm going to be focused primarily on our lead program. This is a single agent that can be dosed every 6 months or twice a year, potentially for the treatment of wet AMD. It's developed off a platform technology that came out of the Johns Hopkins University. And we've also developed a library of compounds that I won't have time to discuss today, but hopefully you can talk about next year that works in our platform as well. So I think we've heard a lot today already, especially on the last panel, about the treatment burden here. So it's obviously a very large unmet medical need. The longer we can increase the duration of response to treatment, the better off it is for the patients and obviously for the clinical practice. We've already done our research and know that it's very strong demand for, I think, optimally a 6-month formulation, which is why it's our focus. So the way we've done this is by taking a known compound, Sunitinib, which is actually Sutent. It's an approved drug that Pfizer got approved in 2006, and we've incorporated that into our PLJ platform technology. And once it's injected into the inferior vitreous, it actually forms an aggregate inside the vitreous of about 5 millimeters in diameter. So it forms an implant that's not disturbing the visual axis, and then it slowly degrades over time. You can see from a single injection in the graph on the far left that the drug levels are achieved at very high levels after a single injection of 1 milligram in a rabbit, and the levels in the RPE choroid and retina are sustained well above the Ki, which is the level that's needed to inhibit all the VEGF receptors, for more than 6 months. And we think this might be the dose that's going to be successfully used in the clinic. Most importantly, you don't see any systemic exposure to the drug, and in this study we only saw a couple time points where there was plasma exposure to Sunitinib. Now, why did we pick Sunitinib? And it actually has a really good reason here, and I'll show you some examples in the next couple of slides. It blocks all the VEGF receptors. So you've heard today a lot about combination therapies, trying to block different ligands. This approach actually allows us to block all the VEGF receptors with one compound, and we think that could be a much more superior approach than trying to combine different agents together. In addition, there's been work out of Johns Hopkins and Don Zach's lab to show that it has neuroprotective properties, so long-term could be beneficial there. And his lab has also shown that it improves photoreceptor survival. So long-term treatment of wet AMD could be beneficial with this compound for that reason. In addition, when you take Sutent or Sunitinib orally, you actually get very high levels in the uveal tract in the eye because of the melanin binding. And unfortunately, you can't give this drug for wet AMD because it has hepatotoxicity, but it has no ocular toxicity that's been reported in hundreds of thousands of patients treated to date with cancer. So Charles Simba, our chief medical officer, was able to find a couple of very interesting case studies in the literature of patients that had wet AMD and also had renal cell carcinoma. The top images that you can see here are basically a patient that had had the wet AMD first presentation, came in, got 3 injections of Lucentis, had some resolution of the fluid, for whatever reason decided not to continue to take the Lucentis, came back about 6 months later with renal cell carcinoma, started taking Sutent. They imaged the retina before and after treatment with Sutent, and you can see after taking oral Sutent, he was actually able to have complete resolution of the retina. So indeed, the drug gets to the target tissues and has its effect when you take it orally. However, it's hepatotoxic, so that's not an option for this therapy. The similar type of finding was also observed in a patient that presented with an ocular metastases from a renal cell carcinoma and also received oral Sutent. Again, the retina resolved after treatment with the oral drug. So we believe the drug will work, and so the key now is to see if we can meet this target product profile, and I'll describe briefly in the next slide the trial design. But the objective here is to achieve comparable safety and efficacy to the existing agents with a twice-a-year treatment. The trial design that we're hoping to initiate fairly soon is a single ascending dose study using patients that are already on anti-VEGF therapy. When they would normally come back in for their next injection of Lucentis, Eylea, or Avastin, we will give them 1 injection of our drug. We will then follow them monthly to look at the durability of the response at each dose level. And then based on that durability of response, we'll select a dose and regimen for the phase 2, and this will be a large phase 2 trial. It's all single protocol, so it'll be the same inclusion criteria for both sets of patients, and it will be a 450 patient trial, head-to-head against the label claim of aflibercept of every 8 weeks dosing or every 2 months dosing. And we hope to be able to share the data from that study with you at this meeting next year. From the phase 1 part of that study, and we'll be starting the phase 2 part of that program about this time next year with a readout by the end of 2019. And then I don't have time to talk about it today, but we're also working on novel glaucoma agents that could be dosed twice a year as well. Thank you very much for your time and attention.